Hello everybody. Today's video is going to be focusing on photosynthesis. Photosynthesis takes place in the chloroplast, and as you can see, I'm drawing some thylakoids, and we're going to be zooming in on this area for further inspection. So here's the thylakoid membrane, it's lipid bilayer, and we're going to have light hit this photomolecule known as P680. And this is part of a process known as photosystem 2. And this energy will cause electron to be released by this molecule, which in turn causes H2O, water, to be split into some protons and then some oxygen. And as this electron is released, its energy will be used for this like proton pump to pump more protons into the thylakoid, therefore increasing the concentration of hydrogen or protons, positively charged protons inside the thylakoid membrane. And then here we have photosystem 1, where light again hits this photomolecule known as P700, and this release electrons will cause again NADP, which is an electron carrier, to be able to carry this electron to be later used in the Calvin cycle. And all this time, as these processes have been going on, there is a definite increase in protons within the thylakoid membrane, and this increase in concentration, as seen here, will be used to drive this protein ATP synthase, as these protons are trying to leave the thylakoid and will produce ATP from ADP and phosphorus. So this is ATP synthesis in the chloroplast, and my friend Chris will be talking to you about Calvin cycle.
Okay, so next we'll be talking about the Calvin cycle. The first step of the Calvin cycle is carbon fixation. The fixation of carbon begins with a 5-carbon compound called ribulose. An enzyme Rubisco catalyzes the attachment of CO2 molecule to ribulose. The resulting 6-carbon compound is unstable and breaks down into a 3-carbon compound called glycosylate 3-phosphate. A single cycle involves 3 molecules of ribulose combining with 3 molecules of CO2 to make 6 molecules of GP. The next step of the Calvin cycle, reduction of the glycosylate 3-phosphate. GP is converted into triose phosphate, or TP, using NADPH and ATP. Then the reduction of NADPH transfers hydrogen atoms to the compound, while hydrolysis of ATP provides energy. And then each GP requires 1 NADPH and 1 ATP molecule to form a triose phosphate, so a single cycle requires 6 of each molecule. The third and final step of the Calvin cycle, regeneration of ribulose. One of the 6 molecules of TP produced per cycle, 1 TP molecule may be used to form half a sugar molecule. Hence, 2 cycles are required to produce a single glucose monomer, and more to produce polysaccharides like starch. The remaining 5 TP molecules are combined to regenerate stocks of Rubisco. 5, as you can see, labeled as the formula. The regeneration of ribulose requires energy derived from the hydrolysis of ATP. And
that leads us into the stomata, which is an opening in the leaf that is surrounded by 2 guard cells. And the stomata allows for water, both water and gas exchange. So H2O will leave the pore, and O2 will also leave, and CO2 will enter the plant through this pore. Additionally, stomata allow for transpiration to occur as the water comes to the leaves. The water can be evaporated, allowing water to travel up through the stem. Additionally, in order to open the pore, potassium will flow into the guard cell, making it more turgid, and in order to close it, potassium will leave the guard cell, making it less turgid, and thus closing it. Additionally, in C3 plants, the stomata is going to be open during the day and it's going to be closed at night. So besides C3 plants, there's another type of carbon fixation in CAM plants. And so in CAM plants, carbon fixation occurs at nighttime, where CO2 is stored in vesicles until the morning. This process minimizes photorespiration, which is the fixation of oxygen. Plants in dry and arid climates use CAM fixation to improve water efficiency, as they only open the stomata at night, when the water loss through the stomata will be less. Examples of this include cactus, agave, and pineapples. There's yet another type of carbon fixation that occurs in C4 plants. And in these plants, carbon is fixed to PEP by PEP carboxylase, which creates an organic acid. This process leads to increased CO2 being stored in the mesophyll cells. PEP has a high affinity for CO2 and none for O2, thus preventing photorespiration, which is bad for the plant. C4 plants are usually found in hotter climates where stomata can be closed for longer periods of time, and examples include maize, sugarcane, and tropical grasses. So photosynthesis is able to take place because of different types of pigments. And so red and blue light are most absorbed by chlorophyll B, which is the main pigment in the photosynthetic process. Other pigments within the chloroplast also exist, and these include chlorophyll A and carotene, which are both able to absorb different types of light. Yet white light is the best for photosynthesis to occur, as the combination of all the pigments together allows for the most efficient light absorption. Photosynthetic activity can be measured using chloroplast suspended in a buffered solution containing DPIP, which is a dye that can readily accept electrons from the electron transport chain of the photosynthetic process. The transfer of electrons to DPIP decreases the relative absorbance of specific wavelengths of light by a solution that contains this dye. And because electrons are supplied by the light-dependent reactions, the more photosynthesis that is taking place means the more electrons that are being freed up, and thus making the transmittance of a solution higher. Independent variables like light intensity or wavelengths thus can be measured through DPIP experiments.